All right, hello everyone. So today I'm going to take you through just the opening setup and a little bit of the results of the modeling kidney function lab. So you should have you have your direction sheet, but I'm going to kind of model what that's all about, and then you have your answer sheet. Okay? And as we go through the lab today in class, I will provide you with anything you need outside of this video. It shouldn't be much. So what we're going to start with here is we have our cup of water. We have our dialysis tubing, which is soaking right now in water. Okay, dialysis tubing is able to open and then it's semi-permeable. So I'm going to leave that in there for just a few seconds. I already tied off one end. We want it to function like the kidney as if we were looking for diffusion. We have our simulated blood. I also have string for the other side. And then we have salt test strips. Okay, and we'll be testing that as we go through. So what we're going to do is we're going to open up our dialysis tubing. And like I said, I already tied one end. And what we're going to do is put about 10 milliliters of simulated blood in here. I'm just going to show this down a little bit so you guys can see it. You don't really need a full 10 milliliters. We'll put in, I don't know, maybe we'll put I'll get away with like 5 today. Okay. Now what we're going to do, tie off the other end. Much easier to do this with another person here. Okay, we'll tie that nice and tight. Like I said, this is a semi-permeable membrane. If you've never used dialysis tubing, it's quite interesting. It looks like a plastic bag, but really it is, it's got little pores in it to allow certain things. Okay, so then I'm just going to, I'll use this water. I'm just going to rinse this a little bit just to make sure there's no blood starting on the outside of it. And that might ruin our results. Okay. And we're going to take that. We're going to put that inside of our cup. We're going to see really how our kidneys filter certain things out. Next thing we're going to do is take our salt test strip. We're going to dip it in. We're going to let that sit for about a minute or two. And what we want to remember here is that our kidneys are semi-permeable as well, right? When it runs through the when the blood runs through the glomerulus, the red blood cells and larger proteins, they don't filter out into the Bowman's capsule as filtrate. And this is going to mimic what would be filtered out of the blood into that filtrate. And then obviously, as we move through the nephron, certain things are able to pass back into the bloodstream. But this is just showing really how this would be done initially in the glomerulus. All right. So this usually gives you a pretty quick sign. So I'm going to ask you to take my word for it. But you can see here that this stays yellow. And if we read through the directions, any change to green tells you that it's turning positive for salt. So your regular water coming out of your tap shouldn't have salt in it. And that's obvious here. So this is going to stay yellow. So that gives us a negative result. Now, I ran this earlier on another solution. And after approximately 30 minutes, this is what we start to see. Okay. So after 30 minutes, I tested the water again. And I can see that I am starting to get a greener color. Now in your directions, it says that if it turns to a light green or a partial green, it tells you you have a low salt concentration. And if it turns to a dark green, it means you have a high salt concentration. Okay, so this is after approximately 30 minutes. You can see it's starting to go green. Not exactly super dark, but starting to get there. And then it tells you to look for your 24-hour mark. And I'm going to let you kind of guess, because on your lab sheet it says to predict after 24 hours. And then at some point today during the lab, I'll give you the answer to that 24-hour mark, or we'll look at it tomorrow. Okay, so as we fill out this sheet, it tells you your observations and what we see. Okay, and I don't have the cup for after 30 minutes, but you can start to see, in there, the cup is starting to, the color in the cup is starting to turn a little bit yellow. So that means that something is filtering out of the blood as well. So after 30 minutes, this outside cup would also be yellow in color, which tells us that things are able to come out of the bag in terms of filtrate. Remember, the filtrate is what is going to be destined to become urine as it moves through all of the tubules and then out the collecting duct and into our calyx, renal pelvis, and then our ureter before hitting our bladder. Okay. So hopefully this gives you a general idea of the setup. Like I said, the original test was yellow for salt. After 30 minutes, we start to see this lighter green color. And I don't know if you can really see it, but the color, the water color is actually starting to turn yellow already for that 30-minute test for the color of the water on your data sheet. So make sure you fill that in, and then you can start answering the questions, and I will give you a little bit more piece of information while we do the lab.